Okay, good evening, everybody. Welcome to this Retina UK webinar. This is one of a series of webinars we are hosting, and we'll be delivering at least one on a different topic each month. We are really pleased to have with us today Michael Gilhooley. Michael is a clinical lecturer in the Department of Genetics at University College London, the Institute of Ophthalmology, and at Moorfields Eye Hospital as well. His research interests include the development of novel therapies for inherited retinal degenerations, so optogenetics, which is what we're here to hear about this evening, and inherited optic neuropathies. Michael will introduce the technique of optogenetics and its potential for development into a treatment for IRDs. We're also joined this evening by Kate Arkle, our Research Development Manager, who will be collecting questions throughout the presentation. So there are a couple of ways that you can ask questions this evening. You can either type them into the Q&A box, and you'll find that at the bottom of your screens. These questions will be asked by the team on your behalf. Alternatively, you can raise your hand. If you're on a computer, you can use the Alt and Y key if you're on a Windows computer, or Option and Y if you're using a Mac. We'll then be able to ask you to unmute your microphone and you can ask the question in person. So please do leave your questions throughout the presentation. We'll have as many of them answered as we possibly can at the end of the presentation this evening. If we're not able to get to your question, then we will endeavour to get those to you, the answer to those over the next couple of weeks. So thank you once again for joining us. And without further ado, I'm delighted to introduce Michael.
Evening, everyone. I'll just share my screen and get started. Right, so good evening, everyone. First of all, I'd like to thank Matthew Carr and everyone at Retina UK for asking me to speak this evening. My name is Michael Gilhooley, and I'm a clinical lecturer in ophthalmology at University College London and Moorfields Eye Hospital. But as you can probably guess, I'm not originally an East Ender. I'm from Ayrshire, but I've been down south for many years working at Moorfields. A bit about my background. I started as a junior doctor in Glasgow and moved to train as an ophthalmologist in London. But around 5 years ago, I had a change of direction and made the move up to Oxford to join Professor Down's team at the Oxford Eye Hospital for a period of research. And in that time up at Oxford, I specialised in the new technique of optogenetics. I've since moved back down to UCL and to Moorfields as a clinical lecturer, and this means that I spend 50% of my time in the eye clinic, the NHS genetics clinic, seeing patients with inherited retinal degeneration and inherited optic neuropathy, and the other half of my time in the university in the labs researching and teaching new ophthalmologists. So in structuring this talk this evening, I thought it might be useful to briefly touch again on what actually happens to the eye during the inherited retinal degenerations, things like retinitis pigmentosa, and how this can offer opportunities for new treatments. We'll touch on the concept of surviving cells or survivor cells during retinal degenerations. And then I hope to tell you a little bit of the story of optogenetics. What is it? Where did it come from? And why is it important? But more importantly, given the recent news of the last few months, how do we deliver this as a technique into an eye as a potential treatment? And finally, we'll have a look at some of the questions that still need to be answered in the lab and the research clinic before we move this technique on into patients as a more routine treatment. So as I mentioned, first of all, I thought it'd be useful to recap briefly what happens to an eye when retinal degeneration occurs and some of the opportunities this process gives us to develop new therapies. As you remember, in these diseases, a mutation or coding or even a spelling mistake in a gene leads to the death of the photoreceptor cells. These are also called the rods and cones in the back of the eye. And these are the cells in the retina that actually convert light that falls onto them into electrical nerve impulses that can be understood by our brain and actually allow us to see the light that comes into the eye. Now, rods and cones are essential to our ability to see. They are highly specialized cells that turn light into something our brains can understand as electrical signals. And targeting these cells, and particularly those cells that support them and surround them, has been highly effective so far in the search for new treatments for inherited retinal degenerations. The example in point is the drug Luxturna, now available in the NHS, which works by supplying a good copy of the RPE65 gene, that's a copy without the mutation or spelling mistake, to the RPE cells that support these photoreceptors and so prevent their death in a particular kind of retinitis pigmentosa or Leber's congenital amaurosis, where the gene involved is RPE65. And this therapy, and several others very much like it that are in development, have been termed gene replacement therapies. What they do is they replace the faulty genes with a good copy. And these have a great promise for the specific inherited retinal degenerations or the specific genes that they aim to treat. But the scale of this challenge becomes apparent when we look at the pie chart on the right-hand side of this slide. This large chart is split into lots of little slices, and each slice represents a single gene that causes an inherited retinal degeneration. And as you might be able to see, there are quite a lot of them. And in fact, as more and more are discovered, we're getting up to around nearly 300 genes that are implicated in causing inherited retinal degenerations. And this approach of gene replacement therapies would require a different drug to be developed for each of those mutated genes, which is a challenge, but something that may be possible. But what we must also take into account is that the largest slice of this pie, at around 30%, is labelled here unknown mutation causing retinitis pigmentosa. So even on a good day, if everyone with an inherited retinal degeneration came to the clinic and underwent genetic testing, there would still be perhaps up to 1 in 3 people or so, where with our current knowledge at the minute, we couldn't yet give those patients an idea of which exact gene is causing their condition. We couldn't give them a molecular diagnosis. And this is one of the drawbacks of traditional gene replacement therapies, is that if we don't know the genetic problem, we don't know which gene it is that's causing the inherited retinal degeneration, we can't construct a genetic solution to treat those patients immediately. Therefore, there's quite a large unmet need for an alternative therapeutic approach, which could be used regardless of what the causative gene is, or even for patients where that gene isn't actually known, where our diagnostic abilities haven't quite caught up with where things are. And this is where techniques that are centered on survivor cell stimulation have the best and greatest potential. So I often find analogies are helpful in describing complex diseases such as inherited retinal degenerations. And one of the favorite analogies of the ophthalmologist is to compare the whole visual system to how we record films. So in that analogy, the eye acts like a camera with lens and film, and an optic nerve, which acts as the cable connecting the camera to the video recorder that is your brain's visual centers. And in this analogy, the retina is often described as the film in the camera. The lens focuses the picture that comes into the eye onto the retina, the lining of the back of the eye, and the picture's developed in the retina. And this is good to a point, but it leaves out a particularly important part of this system. There's actually central when we consider treatment approaches to the inherited retinal degenerations. The retina actually does much more than simply convert light signals into electricity, develop pictures. There is also a huge amount of processing machinery actually within the retina that begins to extract interesting information from what we see. For example, before the nerve impulses have left your eye, within the retina, the interesting information such as the edge of tables or steps, movement in a picture, and where different colours are in a picture are already extracted from the image before this gets anywhere near the brain, where it's traditionally thought all this processing may have occurred. And why this is important comes when we consider the inherited retinal degenerations. as we've touched on, in these diseases, the mutation or coding mistake in the genes leads to the death of the photoreceptor cells, the rods and cones. And those are the cells that actually do the converting from light to nerve impulses. But what is not lost in these conditions in the inherited retinal degeneration is the so-called neuroretina. And these are the processing cells that actually make up about a third of the thickness of the retina that are unable to sense light in their own right. What they do is they process the electrical signals that come from those rods and cone cells before passing them on down the optic nerve to the brain. And it's these surviving processing cells that are known as the survivor cells or surviving cells in the IRDs. And as I said, these useful cells remain relatively intact, relatively unaffected by the disease process of inherited retinal degeneration. However, as they have no ability in themselves to sense light, but only process those electrical signals from the now defunct rods and cones, a patient with an inherited retinal degeneration generally loses their vision. However, if we were to artificially generate electronic stimulation of these cells, then we might be able to restore some of that visual perception. And indeed, this is exactly how the retinal electric chip prosthesis that has been used in clinic already, that's how it works. By stimulating these surviving cells with electricity via arrays of electrodes implanted on the retina in response to a computer-controlled camera, a degree of visual perception can be restored. However, in practice, these chips have been limited in their success for a few reasons. The first is that by their very nature, they're artificial, they're made of plastic and metal, and they don't appear to last very well within the eye, at most a few years. And they're limited in how close the electrodes, metal electrodes can be placed together, and therefore they're limited in the resolution that they can provide. Optogenetics, however, could provide an alternative method to stimulate these surviving cells. So what is optogenetics? So in short, optogenetics is the process of inserting genes into individual cells to make them sensitive to light when they would not normally be sensitive to light. However, there is of course a bit of a story here, and it starts in a pond. So green algae are very simple creatures that live in ponds and sometimes in the sea, and they're single-celled organisms, but they have a very useful trick. As these single cells gain most of their energy from light, they've evolved to be able to sense where the sunshine is, so where the brightest part of the pond is, and swim towards that light. So in a way, why they don't see in the same way as an animal does, these creatures do have some form of sight. They're able to move towards a brighter patch of the pond where they have more energy and are able to do better as they're in the sunshine rather than the shade. However, unlike humans, where, as we've seen from that pie chart, literally hundreds of genes and proteins are involved in the process of sight, Professor Nagel, whose picture's here on the left of the side, the screen here, is a scientist who's quite passionate about pond algae, and that was his specialism. And he actually discovered that this process of sight, in inverted commas, this ability to convert light signals into an electrical state in the cell that the cell can understand and use, in these algae, is performed by a single gene. So rather than the hundreds of genes in a human or an animal, there's only one gene involved in the algae. And this gene is called channel rhodopsin. And there's a picture of the channel rhodopsin protein in the middle of the slide as well. And what this protein does is when an amount of light hits, lands on it, it alters the electrical state of the cell in which it's expressed. Now, Professor Nagel is not an ophthalmologist or a vision researcher at all, nor was his close collaborator, Professor Dieseroth, who's on his pictures on the right-hand side of the screen. He was actually a psychiatrist, a psychiatry researcher, who first noticed the potential of this single gene to alter the electrical status of cells. And he was able to insert the alga gene, the channelrhodopsin, into brain cells in his laboratory and use this to control their electrical state, which is an important technique in brain research, by either keeping them in the light or in the cupboard in the dark. And the technique became quickly established in neurology and psychiatry research, but it took a few years more to work its way into the ophthalmology lab. Where you can imagine, having the ability to control the electrical state of cells in response to light can be very important. And if we think back to our survivor cells in the inherited retinal degenerations, for instance, if we could control the electrical state of those cells in response to light, we could perhaps replace some of the function of the lost rods and cones and restore some light sensitivity, a bit like the retinal chip was doing. However, until recently, there was no real practical way to introduce those algal genes safely into the cells of a human and of a living animal. That changed quite a bit with the first generation of gene therapies, those gene replacement therapies, such as Luxturna, which is now available in the clinic, and they may provide an answer. These new medicines are essentially harmless viruses which have had their own genes removed and replaced with that good copy of the IRD causing gene, in the case of Luxturna, the RP65. But this same technique could be used to deliver light-sensitive genes, such as channelrhodopsin, rather than replacing faulty genes, and bypassing that need to have a detailed knowledge of the gene that's at fault. And this is exactly what we and other groups around the world have been doing in the lab, using these adeno-associated viruses that we know are already relatively safe for human use, and using them to deliver channelrhodopsin and other light-sensing genes to surviving cells in retina with inherited retinal degenerations. And at present, this technique is being extensively trialled in mice mainly. But last month we did have the first report in humans, which we'll touch on a little bit later in the talk. And if we consider the inherited retinal degenerations are actually much more common in mice than they are in humans. Around 20% of mice have some form of inherited retinal degeneration. But mice, like dogs and other animals, are probably less visual than humans are, and they rely much less on their vision. So you'd probably be quite hard pushed to tell an IRD mouse family from a sighted one on the behaviour alone. However, when we perform electrodiagnostic tests, similar to those used in humans when we're investigating retinal disease, we can see the differences, the differences that are much more marked in these mice. On the left-hand side of the screen, we have a mouse from a family with inherited retinal degeneration, and we can see that in response to a bright flash of light, all of the recordings are essentially flat lines. There's no response at all to this very bright light that's shown at the mouse, and the mouse just gets on about his business. However, the same mouse, 8 weeks after treatment with an injection of that adeno-associated virus that it causes production of the channelrhodopsin protein within the survivor cells of that mouse's retina, and we can see the same recordings taken from that retina to the same flash of light. Rather than lots of flat lines, we now have multiple layers of the retina where there are spikes and responses to the light that's been seen. So we've had a mouse there that had essentially lost all of his vision that has had a degree of this light sensitivity restored following this injection, which for me was really quite exciting. And what's been even more exciting recently over the last couple of years is the increasing number of channelrhodopsins that have been discovered in different single cell species, mostly algae and other microbes, and also the number of artificially modified channelrhodopsins that have been developed. So with genetic engineering, the basic channelrhodopsin genes can be altered slightly, and this has given us a wide selection of genes that we can use, each with slightly different properties. Some are perhaps more sensitive to different colours of light, others have varying degrees of absolute sensitivity, that they're more sensitive to dim lights or to brighter lights. And as we'll see later on, some channelrhodopsins require really very quite bright lights to be activated, well beyond what may be encountered in a normal patient environment. More recently, however, some familiar genes have been found human retina that have been discovered to be able to act as optogenetic tools. That is, when they're expressed in cells outside of their usual homes, they can also alter the electrical state of those cells, like channel rhodopsin can when it's introduced into cells where it's not usually found. And these genes can include things such as rhodopsin, which is usually found in rod cells, and melanopsin, which is usually found in the ganglion cells of the retina. And these two have the great advantage of a channel rhodopsin that they are natural human proteins, removing some of the potential risk of immune reactions in expressing algae proteins in the human eye. And it's this increasing diversity of optogenetic tools that's of particular interest to me. We now have a wide choice of tools, of genes, of proteins that react to light in quite different ways. For example, I've put 5 of my recordings on the left-hand side of this screen, each of these from an individual cell that's been treated with a different optogenetic tool in response to the same pulse of light. The 2 on the left have been treated with a variant of that algae channel rhodopsin, and the 2 on the left and the 3 on the right have been treated with human melanopsin, each targeted to a different type of surviving cell within the retina. And as you can see, the electrical responses to the same flash of light vary quite a lot depending on which tool is used and what cell it's targeted towards, which gives us 2 real questions that we kind of need to answer in the laboratory before we move into the clinic. Which of these tools, this huge number of different options we have, which is going to be the best for treating patients? But also, where's the best place to target this? Which type of surviving cell is going to be the best one for us to target in humans? And while we've been working on trying to answer these 2 questions, I feel it's becoming increasingly apparent that there may not be a single combination of that is optimum for all circumstances. For example, a high sensitivity tool may be useful for seeing in low light conditions, but a tool that has faster reaction speeds may be more important to detect motion. Therefore, in the longer term, there may be an advantage to targeting different genes to different types of survivor cells within the retina, in an attempt to recreate some of the huge diversity of responses that we find in the neuroretina, both in a healthy neuroretina, and hopefully give the patient a more realistic visual experience. And I make mention of some of these questions particularly as an illustration just of how much work is going on and really still needs to be done in the lab. However, earlier this year we had some really quite encouraging news regarding the use of this technique in humans. And that was that we had actually the first report of the technique of optogenetics being used in a human back in May. Of course, the BBC News take might have been slightly more dramatic, with algae proteins partially restore man's sight, than perhaps the more measured article title in Nature Medicine, which was the scientific article that was published, and it says partial recovery of visual function in a blind patient after optogenetic therapy. And while I was hugely excited by this report, we do need to inject some caution, as we always have to do in science. The main reason is that we don't have that much information about the particular study. This article describes actually the experience of a single patient in France with an inherited retinal degeneration who had very poor vision and was treated with optogenetic therapy as part of a safety trial. So bear that in mind. Although this is the first time this has been reported, which is hugely exciting, it's still only 1 patient. So we're still eagerly awaiting the reports of the other patients that were treated in that study. Now, the study was entitled Pioneer, and it recruited 15 people between 3 places, France, the UK, Moorfields, and America. And each of these people that were recruited into the study had an advanced inherited retinal degeneration with very poor vision. The best vision they could have would be perception of light. And these 15 patients were given an intravitreal eye injection of a new drug that was called GS030-DP. So I wonder if they might rename the drug if it does come to being used in the clinic, because I find it a bit of a mouthful. And they treated these patients with different doses. The main aim of this study is really to establish that the drug is safe, that it doesn't cause any untoward reactions, and also to work out what the optimum dose is. And GS030 is indeed like Luxturna that's already licensed for LCA, an adeno-associated virus, but it contains a modified channel rhodopsin gene rather than a replacement gene. And the gene it contains is something called crimson R, which is a kind of channel rhodopsin from a slightly different type of algae. And it's called crimson because it's more sensitive to red light than the green light that's preferred by the pond algae, because the ponds are usually green, I'd imagine. So this drug would enter the eye by the injection and introduce a copy of this crimson R gene into the survivor cells of the patient's degenerate retina, with the aim of making those surviving cells sensitive to light in their own right. And in this study, demonstrating the safety was particularly important, given, as we've touched on before, that crimson or channel rhodopsin is not a human protein. It's not a mammal protein. It comes from an algae, which is very distant from humans. And there's a chance that the patient's immune system could recognize this protein as not being part of them and begin to attack it. However, the secondary endpoint in this study was to demonstrate the efficacy. Did this treatment actually work? Did it restore some kind of sight to this patient who had very poor vision? And the choice of the crimson R variant of channel rhodopsin was particularly useful here when we consider the point of the study, which was to work out the safety side of things. One of the disadvantages of channel rhodopsin is that it's one of those proteins that requires very, very bright lights to be activated. And in planning this early trial, this disadvantage was kind of turned on its head. And they argue that if there were to be a safety problem with the treatment, the fact that the crimson R would require very bright lights to be activated could be protective. Essentially, the patient would need to stay away from very bright lights, which hopefully we'd all do anyway, in order, that's all they'd need to do to stop the channel rhodopsin being active. This did, however, mean that special amplifying goggles had to be worn during the trial. So these used a computer and a camera to reproduce the outward scene that the patient could see, but amplified the brightness many, many fold. So I don't think they were particularly comfortable to wear and perhaps slightly cumbersome, but useful in this study, given it's the very first time this kind of treatment's been used in a human. And we'd hope perhaps in future trials we would perhaps use a tool that has a higher sensitivity, needs much less bright lights to be activated, so that we wouldn't need these goggles. And why the article does not report in detail the efficacy data or the safety data for all the 15 patients, it's really only from this one gentleman from France. We assume that they're doing that because they're putting together an application to have the drug approved, or the efficacy of this, the information for this one patient is reported in a bit of detail. So following his injection, as part of an 18-month program of testing that went on from his injection for 18 months afterwards, 3 main measurements of his visual function in the treated and untreated eye, both with and without his stimulating goggles, were made. And these were visually guided tasks. So his ability to find an object that was put on a table in front of him without any other cues other than his vision, to count and locate beakers on a table, and to perform the same task while connected to an EEG machine, an electroencephalogram. And this is recording his brain waves from the different areas of the brain, with particular interest in the electrodes, the recordings from the back part of the brain, which is most intimately involved in the processing of vision and how we see. The idea there being that to prove that the visual information is coming from the eye and arriving in the back part of the brain, where it be able to be processed and to be able to alter behaviour. But I thought, rather than going through graphs and reports from those individual things, I thought it might be the easiest way to demonstrate this, I think, is just to observe this gentleman performing the tasks he's been given with the goggles turned on and off. So here, this may be a folly to try and use a video during a lecture, but I've managed to get hold of a video of this patient actually performing these tasks. Now, as we said, he is French, so the video is in French. There are subtitles there, but I'll explain what's going on in English over as well. But it's we'll just start and play that just now.
Pourtant, je balaye bien, je pense.
Essentially, he doesn't see anything. There's a black object in front of him on the table. That's with him with none of the stimulating goggles on. He's not able to see the quite dark black object that's in the white table in front of him. The second video, he has the eye that's not been treated. He only had one eye treated as covered. He's got the treated eye open, the one that's had the injection, and he has those light stimulating goggles on to amplify the light signal that's coming into his eye. So this video, he's put the goggles on and he's having a look around the table. There's no audio at this point. He's just fiddling with the glasses for the first few seconds.
Là, j'ai quelque chose.
Here's that here. I've got something.
Qui ne paraît pas être le... Non, c'est ni un bord ni le haut, puisque ça me donne le sentiment d'avoir une espèce de vibration un peu verticale.
It's not the edge of the table, but there's something that's vibrating or shimmering.
Très blanc. J'ai un truc qui vibre verticalement.
He's reaching out his hand and he grabs the object. So as you can see, that the level of vision that's returned to this gentleman, it's not photographic. He's not able to use it. It's not giving him a complete restoration of his vision. But this is a gentleman who before had in that eye not a perception of light. You know, he's completely dependent. He couldn't see anything at all, and now he's able to use the sensations that he's getting from that treatment to perform a visual task. He can locate the object on the table. And it's also interesting the way he describes the sensations. They're not as pictures, but as vibrations. But he is able to use that information in some way. It's getting to his brain in such a way that he can pick up the object and see things, which to me, I think is quite impressive for the first time we've ever used this treatment, and given that this gentleman had such poor vision beforehand. So while we've now demonstrated that visual responses can be returned in theory in inherited retinal degenerations, there are several outstanding questions that need, I think, quite a lot of research to improve this technique and really make it as useful as it can be for people, for patients with low vision. And those questions were already touched on, but which is the best tool to use? Channel rhodopsin or crimson R is one option, but there are many others and more coming online all the time, from algae, animals, and artificial ones that are beginning to be created in a lab as well. The second question is, where do we target these genes, these tools? Do we try and get it into as many of the surviving cells in the retina as possible, or do we choose a particular type, ganglion cells or bipolar cells, etc., that might be more effective if we're more exact in where we target the treatment to? And then thirdly, what a lot of us are waiting quite eagerly for is the report from this pioneer trial, that really the safety data, the systematic safety data. These are algae proteins, they are non-human proteins. In theory, there's the immune system could be activated and begin to attack these proteins. So this is a very important question that needs to be answered. Are these completely safe? And if they're not, other things we could do to mitigate that risk as well. And that hopefully will come fairly soon with the results of this trial. And then finally, the usual full clinical trials will be needed. This is hugely impressive data that we've had from this publication earlier in the year, but it's still only from one patient with one particular form of inherited retinal degeneration, treated with one option. How does this compare to both other patients that have already been treated in that trial, the other 15, and how will it compare to a more systematic, larger clinical trial? So I hope I've been able to give you a brief review of the disease processes in inherited retinal degeneration, the concept of the survivor cells as an avenue for potential novel therapies, an introduction to the concept and the story of optogenetics, and perhaps most importantly, the questions that remain in this area as we move with some speed really towards its use in patients. I'd of course be more than happy to get any questions both now or if anyone wants to drop me an email later on. But most of all, I'll just say thanks for listening this evening.
Michael, thank you so much for that. That was absolutely fantastic. I always look forward to hearing you speak. It really is exciting, some of these potential treatments and things that are coming. The research is phenomenal. So we're going to move into the Q&A section of the evening. So I know we've had a few questions come in throughout the presentation, but if you've not left your questions so far, again, there are a couple of ways you can do that. You can either type your question into the Q&A section at the bottom of the screen, or you can raise your hand and we'll ask you to unmute so you can ask the question yourself. So just to remind you how you can do that, you can press the Alt and Y key if you're on a Windows computer, Option and Y if you're using a MacBook, and if you're on a tablet, if you go into the reactions section, you can then raise your hand from there. So I'm going to hand over to Kate Arkle, our Research and Development Manager, to do this next session.
Thank you, Matt, and thank you very much, Michael. That was really, really interesting. I always think optogenetics, it's such an ingeniously simple idea to use these survivor cells and give them the instructions to take over. But actually, your talk was a really good illustration, I think, of how complex it also is, and that we've got all these options, and we're really just at the beginning of finding out the best way to use this amazing tool that we've got potentially in front of us. So that was really, really good, and we've had some really good questions typed into the Q&A box. So, but we welcome more if anybody thinks of some. So the gentleman you showed in that video was obviously at a very advanced stage of sight loss, and somebody's asked, is the focus broadening yet so that optogenetics is not solely focused on those with complete blindness or near complete blindness, but that it could be developed to boost the vision of those whose vision loss isn't quite so advanced? So, I mean, I guess that's perhaps on its own or as an adjunct to another treatment.
Yeah, I think that's a fantastic question, and I sort of split that into two ways: what's actually going on in the research clinic and in the lab, what's actually in humans. And I think at the moment, it is going to be for the next few trials, I'd imagine, in people with very poor vision for two reasons. The first, that safety aspect, that actually we need to make sure this is absolutely safe. The one thing we would never want to do is introduce a protein like channel rhodopsin into a retina that's already damaged by the inherited retinal degeneration and then set off the immune system that might actually make things worse. Now, that's our question at the minute, because we're just going into humans. I'd temper that though with where things are in the lab a little bit further back. As I mentioned there, a little bit of what I do with my family of mice that have an IRD, and things are obviously a few steps ahead there, and that's exactly kind of where we're getting to begin to ask those questions. It's been done for a few years now that we've treated mice that have very poor vision, and some of the questions are beginning to be asked now: is at what point would be the best time to actually give this treatment? So is it best to wait till the end, till all the natural vision's gone? Or is it best to intervene at an earlier point? And that's something actually I know in Oxford, one of the groups, they're actively looking at that, treating mice at different ages to see actually if you could capture it early, does it prevent the mice losing sight? But as I say, I need to temper that. In humans, it's probably a few years off, but actually that's the direction things are heading in the lab as well.
That's really exciting to hear, because I think that question is thought of by a lot of people actually. And do you see it being used alongside perhaps another treatment at the same time as well, potentially?
That, in theory, yes, that's one of the great advantages of it. That if say you had a molecular diagnosis, something RP65, and actually you were eligible for that treatment, but all those the gene replacement therapies are going to do is stop the ongoing process. It won't reverse what's already been lost, and that's the the long-term hope of something like optogenetics is it might be able to actually improve the vision there. But I would say that that's well down the line. But that's in theory one of the great advantages of optogenetics over the first generation of gene therapies.
That's really good, thank you. I'm just looking through the other questions. Well, actually, this is kind of a linked question, I guess. So is it the case that optogenetics won't work as well as gene replacement therapy? Because in optogenetics, you're using cells that aren't photoreceptors or whenever. So
yeah,
kind of
yeah, I think you're absolutely right. I think that's one of the drawbacks to it. Will you get the same visual experience from something like optogenetics? And I think the answer is, in humans, we just really don't know. We know that our brains are plastic; they can regenerate and adapt very well to things. But whether they'll be able to adapt to the different signals they get back, which is why I think it was very interesting. The man that had been treated, there's a slightly longer video there that you can look at the Nature website. He doesn't describe what we would understand as vision. You know, he doesn't see a black or white things. What he says is he often describes it as shimmering or an impression that there's something there, and in that way he was able to use whatever information he's getting to reach out and hold the object. He was able to use the information he was getting from his eyes, but he wasn't describing it as a perception in the same way. So that is going to be very interesting as we go forward, as more people are treated. How does it actually feel to be treated with this? Does it feel like your vision's come back, or does it feel slightly different, but you've got enough information to navigate around a room or recognize people? I think that is something that we just can't do in mice. You know, you ask a mouse what it can see, it's not too bothered whether it can see or not. But yeah.
Yeah. There's another question that actually follows on quite well as well, in that perhaps about background, is that this person says they understand that optogenetics is gene agnostic. So what we mean by that, it can potentially be given to lots of different people, no matter what their underlying genetic fault is. But do you know yet if it will work better for some IRD causing genes than others? And if so, which ones?
In terms of hard facts, I can't tell you yes or no. That's not something we've tried. My estimation would probably be, the answer to that would probably be yes. There are some mutations like LCA-RP65 where the genetics is actually very well understood. It's a recessive disease, so all we need to do is replace the gene. There are other genes where the genetic side of things is actually much more complicated. Those dominant, autosomal dominant diseases where the mutated gene actually makes toxic elements, and how an optogenetic treatment would interact with that will be much more interesting. It could be that in those some conditions, when you have something like RP65, it may be that gene replacement is the best option and is adequate for a lot of people. And that's well and good, but it's a long time before we're able to be actually to compare those. But I would imagine, yeah, that we'll probably at some point get to a point where you'll come to the clinic, you'll have your genetic testing, your genetic diagnosis back, and then you have this gene, we'd recommend this particular treatment off the shelf. And that's, yeah, hopefully in 20 years time, that's what we'll be doing.
Yeah, I think these forward-looking questions are really just testament to the amount of excitement that this potential has generated, really. I think our community can really see that this has got hope for so many of the community. So another good question that actually comes up a lot, do you think it'll ever be feasible to have the therapy without the goggles, without the eyewear, as it gets more refined?
Yeah, I think that one I can answer. I think absolutely yes, and I think the reason for those goggles is the Crimson R, the treatment they used in that trial was one of the first channel rhodopsins that had been modified in any way, and that was perhaps actually in the lab, maybe getting on for 10 years ago or so now. In those 10 years, the number of tools and the modifications we can make to them have advanced hugely. So we have newer versions of that channel rhodopsin that are much more sensitive to light. So I think that is a question I can easily answer. I think probably with maybe not the next trial, but coming forward into humans, that will be something that I think we now have the technology in the lab to fix. But actually, I think it was a good point they made in that paper. One of the reasons they chose that less sensitive treatment was if it didn't work or caused damage, they essentially kept this patient away from very bright lights. And when I say very bright lights, the light that comes out of these goggles is kind of like football pitch floodlight that
Okay.
if you were fully sighted, you find it very uncomfortable to wear them. Yeah. Yeah, so that absolutely, I think that's a blip in terms of this initial trial.
That's really good to hear, because I know, I mean, they look really cumbersome, those particular goggles, and I think, but in reality, that's a lot of weight for somebody to wear.
Yeah, it's not a practical way to.
So yeah, really great to hear that's within reach. Is optogenetics, another question we've got about, is the number of injections? Is it a one-off treatment in the same way that, say, Luxturna would be?
Yeah, in theory, yes, it would be a one-off. In the same way as Luxturna, it's just the gene you're replacing is not your RP65. It's just you're introducing this channel rhodopsin gene, and your own cells would make it continuously in the same way as the
Honestly,
Luxturna.
Yeah, it's how long that treatment effect would last.
That's a really good question, and I think that's something for Luxturna as well. We won't know until people have been treated for 10 or 15 years how long it'll last. In theory, at the genetic level, in theory, it should last for a long time. You know, as long as the cells are alive. But practicalities, we just don't know, I'm afraid.
Yeah. Another interesting question. What are the impediments to using human photoreceptor proteins rather than ones from algae? I guess, is it just that the algae ones came first, that we understood them?
Yeah, this is my pet project. Well, and that's the group in Oxford. If anyone goes to the Oxford Eye Hospital, Professor Hankins and Professor Downs there, that's really one of the things that they pioneered in Oxford, is this melanopsin especially, which is a human protein that's found in some of the cells in the eye anyway. And they've done a lot of work in using that in the same way as the channelrhodopsin, introducing it into other cells as an alternative, and the idea there is that there's no argument in terms of the immune reaction, because it's a protein that's there already. The immune system won't bother about it. It has its own advantages and disadvantages as well. It's much more sensitive than channelrhodopsin, you know, because it's at human light levels, you know, so you'd use it in your normal vision. But one of the disadvantages of melanopsin is it's very slow. So its main function in the eye, in a healthy human eye, is to regulate your sleep and wake cycles. It's not actually involved in sight. So people are often amazed by this. The eye does other things other than see. But one of the most important things is it regulates your circadian rhythms and controls your pupil and other things in the eye. And melanopsin's developed to be a sort of on-off switch. It's not quick. So you don't sort of wake up and fall asleep whenever you turn the lights on and off. But when the sun rises or the sun sets very slowly, that melanopsin is adapted to that change. So that is one of the drawbacks of it, and we may have to make some modifications to speed it up so that you don't end up with just sort of essentially still pictures and then close your eye, another still picture, that you can actually have motion. It would be like a film rather than a still picture. But yeah, that's absolutely something that's been worked on, but it's a few years behind channelrhodopsin because it was the first one on the scene, so it's moved through the most quickly.
Yeah. This is really, I'm learning loads. I don't know about all our attendees, but this is fantastic. There's two sort of slightly linked questions, and that somebody's saying, is it possible to have an appointment to see if my father would benefit from the treatment? Now, I guess this is sort of leads on to the other question, which is who's doing clinical trials. Somebody else has said Bionic Sight and GenSight have both had early successes. I think that was GenSight, wasn't it, that you showed the video from? Yeah. Are there any other companies in the area? So I guess what's going on in terms of clinical trials that people could actually potentially access?
Yeah, so certainly the main ones are those GenSight ones, and I think the main advice is, as always, is if you have an inherited retinal degeneration or have a relative with them, make sure you're in the local genetics clinic if you'd like to go ahead with molecular testing. And the main site for recruitment in the UK was Moorfields, the big genetics clinic in Moorfields in London. However, they are interlinked with the other genetics clinics within the country. So if there is this particular kind of patient they're after for the trials, that the call will go out to all those clinics. But the main gen site's been based through the Moorfields Clinical Research Facility, and I think that's just by chance. They were looking for the first UK site, and we have a link. One of the consultants that works in France as well, and it started that. So I think they'll expand from there. But the bottom line is, as always, get yourself to a genetics clinic, molecular diagnosis if it's the right thing for you, and once you have that, you'll be on the databases and pulled into the any clinical trials you'll be offered if that's something that's appropriate for you.
Questions just popped into my mind actually with you saying that. If people have gone for clinical genetic diagnosis and they've had not had a result because the panel hasn't turned up, does that preclude them from because optogenetics is kind of
no, these patients in this trial were some of them. I think they all had a genetic diagnosis because it's a scientific trial, but there's no reason why they would need one. You know, they didn't have the same genetic diagnosis. You know, but the diagnosis within that trial, it was just they had confirmed inherited retinal degeneration. So I think probably in the later trials that come on, that'll not be as stringent a restriction on who's recruited. Definitely not.
Just to clarify, Moorfields isn't recruiting now. That's that's closed.
That 15 patients have been closed, but I believe GenSite will be moving on if they get approval, so that there will be recruitment at some point in the future. When that is, I don't have the details as yet. Yeah,
no, that's fine. I mean, we try to keep an eye on these things too, so I would just say to those people wondering, we'll do our best to keep our information up to date. We don't always get told by these companies exactly what's happening, but we do our best. But also Moorfields, if there's going to be a UK site again, I guess it's going to probably be Moorfields, so people can, I think, register at Moorfields. There's a Moorfields research registry of some description as well, which
there is. Yeah, you can find the website's fairly terrible, but if it's on the website, if you look for it in the CRF, which is the Clinical Research Facility, the other way to do it is speak to your own ophthalmologist and have them refer you to Moorfields. You don't actually physically need to come to the hospital, but just they make us aware of Moorfields that you exist out wherever you are. But,
fantastic. Okay, just looking through, we have actually got somebody with their hand up who is on the phone. Matt, is that just the same process? Excuse me, just one moment. If someone's on the phone,
Oh yes, hello, can you hear me?
Hello, yeah, I can hear you.
Yeah, I have a question. Would this kind of research and treatment help be beneficial to people who have Usher syndrome?
In theory, as much as it would be for any other inherited retinal degenerations. So absolutely, yeah, it would apply to Usher's. Yeah.
Oh, that's good. Thank you. Thank you for answering my question.
Thank you very much for sticking your hand up and asking. We've got another one that I've just found at the bottom of the Q&A box. Will people that get this type of treatment still be able, we have kind of all sort of answered this, but I still think it's worth just specifically addressing, still be able to get gene correcting treatments later on? Will there be an a problem with the immune response with the AAV vector? Basically.
That's a really good question, and I think I've kind of conflated this a bit. There's kind of two immune issues. One is the drug itself, the virus, and one is the gene or the protein that virus expresses. And there's a sort of ongoing debate about the virus side of it. Things like Luxturna. If somebody was treated with Luxturna, if we find out in 10 or 20 years it only lasts for 10 years, will we be able to treat that patient again with the same treatment, with the same virus? Because once that virus has gone into your body, if the immune system's caught it, the first time it might not causing a reaction, but a second dose, a bit like your booster dose for your vaccine, you know, if the immune system sees it again, it may set off more of a reaction. And there's a lot of debate in that in all gene therapies about how permanent it is, what the reaction will be in the longer term. The other issue with the optogenetics is the actual protein that's going in there. If we're using a non-human protein, the same thing will apply. If it returns again, will that induce an immune reaction? And that's something we're actually looking at in mice, not so much the virus side of it, but the payload, the channel rhodopsin reactions actually are the particular reactions within the retina. And that's something we've got a colleague who used to work with in Oxford's moved back to Germany, and we're doing a project there funded by Pro Retina Deutschland, which is the German equivalent of Retina UK, are funding a good bit of that research as well. And we're trying to look to see actually, are there immune reactions that are specific to the channel rhodopsin or the melanopsin, independent of the virus, and is it going to be something particularly about optogenetics that is more immunogenic? It causes more problems with the immune system than just straightforward gene therapy like Luxturna.
Perfect, thank you. Let me just look through. Good question. In addition to optogenetics, what else do you think might be a game changer for IRDs in the coming years?
I was going to say optogenetics is my thing, but I think the thing with optogenetics is it has huge potential, but it's still, you know, to be a off-the-shelf kind of treatment, that there's so much science still to do. You know, channel rhodopsin was only discovered, you know, 15 years ago. This is moving at pace, but there's a long way to go. I think the gene replacement therapies, although they won't be applicable to everyone with IRD, we've got one in terms of Luxturna, there are others coming up behind it. I think in the short to medium term, that's the game change that we're already at the beginning of, I think is really going to alter things. And seeing how they work in the long term, it's going to be really fascinating.
Thank you, Michael. I think that is a nice question to close on. Somebody's just said wonderful work and presentation, so thank you very much.
Thank you everyone for listening.
No, it has been really interesting, and thank you all of you for some really good questions there as well, which I just think has really helped to round it out too. So thank you very much. I will just briefly, I think we're almost bang on 8 o'clock, so I'll just briefly hand back to Matthew and we'll close up.
Thanks, Kate. Thanks, Michael. That was fantastic. Those questions were amazing. So yeah, huge thank you, Michael. We really value your presentations when you come along and speak to us. And of course, thank you to everybody else who's joined us on this evening's webinar. So as mentioned at the beginning of this evening's sessions, we are planning to host at least one webinar each month, and details of the next will be available to you shortly. So just a reminder that Retina UK is a registered charity. We receive no government funding, and we rely on our wonderful supporters, such as yourselves, to raise the funds needed to provide the vital services and invest in groundbreaking medical research. So if you are interested in supporting our work to enable us to do more webinars and things like this, there are so many ways that you can get involved, from a sponsored walk, a bake sale at work, or just from a straightforward donation. So please do reach out to a member of our friendly fundraising team if you wish to support us further. You can do that by emailing them on fundraising@retinauk.org.uk. So just following this webinar this evening, we will be sending out an email at the beginning part of next week, which will have details of where you can rewatch and listen to Michael's presentation, and details on how you can book onto our other events. We'll also be seeking your feedback on the session today. We do value all feedback we get, and yours will really help us to develop our webinars and our other services. So once again, thank you ever so much to Michael, and thank you to everybody that's joined us this evening. And we wish you a safe evening. Thank you very much.